Good morning, everyone. Hope you had a good day yesterday. Today's going to be another good day, hopefully. I have a couple of announcements before we start the scientific program. The first one is that Alex Pentland, one of our keynotes and the chairman actually of this morning session, has made a very nice gift to the conference. Brought some of these very nice books with him for all the students, participants. So whoever wants to get a free copy of this, and is a student, so please identify yourself. If they are, I mean, some of the guys I see here go there. I mean, they will not buy it. They have a list and the names and everything. They can get a free copy then from the reception desk. The books are there. They just got here yesterday. We're supposed to put them in the bags, but they got stuck for 3 days at the customs. This country is very serious about taxes, so we had to make a few phone calls and so on, but now they are in-house. Second announcement is about tonight's dinner, and most importantly, the boat cruise, which starts the whole program. The cruise starts at 6:30 p.m. from the Market Square, which is the same place where City Hall is, the same square that you've seen, most of you have seen yesterday, where this nice building where we've been yesterday night. Of course, the port is like a couple of 100 meters long. There's usually not so many people, especially at that time. There will be many orange T-shirts just to indicate the 2 points where the 2 boats, because there were too many for a single boat, are going to leave. So try to identify those orange T-shirts. It should be fairly easy. If you want to know the exact point where the boats will be leaving, then you can ask more information downstairs. But I don't think you need to do that. So there will be these groups. Try to be there at least 10 to 15 minutes in advance, because it takes some time to board the people. One of the 2 boats is quite a bit larger than the other one, capacity of more than 200 people. The other one is a bit smaller, because we didn't find large enough boats for this thing. So we'll have to split a bit, but it's going to be social anyway. It's going to be wine on board and so on. Those boats will just make a 90-minute cruise, and then they will land at this island, Klippan Island, where the restaurant is. And we'll have our dinner there, and then there will be further instructions about the way back. But more to that later. And now we can start. So I will introduce Alex Pentland, our chairman of today's session. Thanks a lot.
Great. So thank you all. Wonderful to be here. So I have a really easy job, which is introducing a couple of very famous people. So they require almost no introduction, but I'll do just a tiny bit. So Laszlo Barabasi should need no introduction. He is a famous author of Linked, as well as other popular books, is known for really bringing the notion of scale-free networks to the fore, and recently, last decade or so, setting up a great lab near where I work at Northeastern that spans everything from political science to biology. So you're a true Renaissance man. Laszlo, please come up and inform us. Thank you.
Thank you. All right. So we have lots of things, but none of them gets me across, right? Is this working, by the way, or I have to turn it on? No. It's on. Perfect. All right. Good morning, everyone. Sandy, I did want to mention that I'm forever a student, right? So do we qualify? But I gave this grandiose title, Science of Success, and I would like to start with a more personal story, because that's the one that actually helps us remember things. And let's go back about almost 20 years now, and some of you may be aware that often my claim to fame is based on the paper that we published in '99 with Reka Albert, who was a student in my lab at that time. And when we published that paper about scale-free network, as Sandy has mentioned, we were really very excited about it. We thought that we had some really cool result. I was so excited about it that actually I had to write the paper in 7 days before I went for my vacation. So really, it really kind of captured us. And the way you look at whether it actually captured the community's interest is that you start looking whether there's anybody following up on that, right? And one way to look at whether anyone follows up on your work is that you look at citations. See who cited you. And the first year was a little bit weak, a few people cited it, but granted the paper came out in October only. But then the year after, actually about 100 individuals ended up citing the paper. And that was huge. That was much bigger than I ever did in my life before. So it was a very, very exciting moment. And then we got a huge endorsement. The huge endorsement was that the paper of the century was published. You remember what was that? The Genome Project paper, also one of them in Science. And that science paper, actually, the genome paper, to our great surprise, ended up citing our paper. And they had only a finite amount of citations, and here was among the last one saying the future belongs to thinking about not genes but networks, and there was a citation to our paper. So that felt really good. But then soon we realized actually that despite that endorsement, we are in a very different league. And let me tell you why we're in a different league. Because in the next 2 years, the genome project, the paper on the genome project kind of exploded in citations. As you can see, year 1 collected about 500, year 2 almost 1,000. We were nowhere ever close by. So what I want to actually do now is to talk a little bit about the patterns of these citations, that what determines a citation, and how do we think about how the citations are growing, and what's the difference between two papers. In this case, it's a good control case because they were published in the same journal about 2 years away, so it cannot be a journal-dependent effect. So how do we think about what determines a citation? And the first thing that comes in mind, obviously, is the journal. If you publish in a very high-cited journal, high-impact factor journal, you get lots of citations, right? Well, not so. And let me show you why it's not so. So here I'm going to show you a list of papers with different journals with different citation impact factors from 1.6 to 44. And then let me show you the citation distributions of the papers that they got about a decade after they were published. And what you realize here, I'm showing you 3 journals, Cell, PNAS, and Physical Review B, with very, very different impact factor. Usually Cell has, I don't have it here, about 40 or 50. Physical Review B has about 4 or 5, and then PNAS has about 15 or 12. So hugely different impact factor, yet the number of citations that the papers get are very comparable, except at the tail end where the high impact factor journals get a couple of papers that are much more cited. So when you look at it, the first thing you realize is that even if you land your paper in the top journal, there's a very, very good chance that we'll get no citation ever, right? Or a very, very few. And that's what the distribution shows. Most papers get 1 to 10 citations 10 years after their publication, whether top journal or bottom journal. So the impact factor doesn't really predict the future number of citations your paper will get. Well, maybe the citation count does. So maybe you say, well, impact factor doesn't, but if you are in a certain journal and you got a certain number of citations, that will tell you how many more you will get. Well, here's what we did. We ended up taking a bunch of papers that published in the same journal that after 5 years got exactly the same number of citations and said, what happened then after that? And you see, some of them went on for the next 10 years and get like 5 times more. Others got nothing more. So clearly, just the very fact that you are in the same journal and you get a certain number of citations by a certain time, again, is no predictor what will happen to you in the future. So the problem, let me tell you a few problems that I have with the whole impact factor game is that, of course, it favors established researchers and lacks long-term predictability. And you can see actually the chaoticity and the lack of predictability of different papers if you look at the citation history of papers published roughly at the same time. Here is a bunch of papers published in the same journal and their time-dependent citation, and what you see mostly is a chaotic pattern, right? Completely, apparently unpredictable. Yet, what I'm going to try to convince in the next few slides that this is not so. That these curves really hide a very deep sense of order that is mathematically very, very accurately predictable. And despite the differences, they all follow exactly the same law. And how do we know that? Well, first we're going to make up a very simple model of citation patterns. Question, what determines whether your paper will be cited or not? And the first thing is the quality of paper, right? That's a very fuzzy thing. How do I measure the quality? I don't know yet, but clearly we feel that there are differences, between two papers once we read them. So we have a sense, even though it's hard to quantify. That one I would call the intrinsic novelty or fitness. Then there is the preferential attachment factor saying that if your paper already got some citations, it got visibility, and the more visibility it got, easier to find the paper, hence more likely that will be cited. It's well documented in the literature. And finally, there's an aging effect, and this is the bad news, that all papers eventually use their citations because the knowledge they presented becomes common knowledge. And we don't feel anymore that we actually cite it any longer. We know that, right? And that aging effect has several analytical forms. This one actually we find to describes very, very well the time-dependent aging in the system. take for now for granted, I'm going to come back how this looks like. Take this one for granted, and if you have this probability that your paper will be cited following this formula, turns out you can write a differential equation and you can solve it analytically, and that will tell us that the number of citations your paper gets in function of the time will follow this ugly function that looks ugly but actually is very simple at its core. Because what it tells us is the following. It tells us that each paper follows a curve like that. That's the citation shape of all papers, no matter where you publish it. May not be easy often to see it, but behind it, it's always there. And the difference between the papers comes in these three parameters, lambda, mu, and sigma. So it's a three-parameter problem. And the first parameter tells you how far you actually ever go. And that's what I call the fitness. You see, how interesting is your paper to your community. The second parameter, it tells you when you're going to reach your maxima. That is, when do you get the most number of citations? And here's the bad news. 99% of the papers reach actually their peak at about 2 years after their publication. And after that, it's just downhill from there. And that is the reason why the impact factor measures the first 2 years of citations only, because beyond that, there's not much happening. It just goes down. And then the third one tells you the decay rate, that is how fast or how slowly the citation decay. And there, the good news is that most papers have very comparable decay rate. This parameter sigma is the same for all of them. So what I'm claiming is that these three parameters describe all citation curves, and let me show you that this is actually indeed the case. So here are the citation curves of 4 papers that were published in the physics literature that obviously have very, very different citation patterns. One of them just has an early peak and decays. The other one is just kind of noisy. The third one actually goes on and gets lots of citations, and the fourth one keeps getting more and more citations every year. These papers cannot be more different from each other, right? Yet, if I'm going to change the horizontal axis of time to a rescale time that my formula is suggesting, and the vertical axis, rescaling it with the fitness of the paper that I get by fitting my formula to these curves, I found that all of them collapse exactly onto the same curve. You see, so they follow exactly the same line. And the only difference between them being these parameters, sigma, mu, and lambda. And not only these 4. You can take on the whole issue of Nature, Science, Cell, PLAS, Physics Review, and Review of Modern Physics. Very different impact factor journals, very different areas covered, and they all collapse exactly on the same curve. So down the line, despite this chaoticity and individuality of the different papers, they all followed the very, very precise law. And the reason they do so is because citation patterns capture the collective interest we have about papers. It's really not about your paper, but it's about how we perceive your paper, how we actually look at it. And that is really a collective phenomena, and that can be captured very accurately analytically. I cannot figure out a priori how good your paper is, but once you put it out, the community already has an idea how good they are, and the citation patterns follow a very deterministic curve telling us what is happening there. Now, of course, you would say, physicists say they give me 5 parameters and we can fit an elephant, right? Or something like that. So 3 parameters is a lot. Do they all matter? And do they all determine your eventual impact? So we came up with the following measure. Let's measure your total impact of the paper. And the total impact paper we defined as the total number of citations your paper will ever get during its lifetime, right? Which is mathematically simply the area under the citation curve over here. And here comes something beautiful out of this. If you take this formula and you take the time to infinity and you integrate it, it turns out that the total impact follows a very, very single curve that depends not on 3 parameters, but only 1 parameter, the fitness of your paper. So therefore, if I go ahead and fit your parameters early on after your paper appeared, at the 3 to 5 years, we can actually get a pretty accurate measurement of what your fitness parameter is. That single parameter will determine how much impact your paper will ever have during its lifetime. And when you reach your peak, as well as how fast you decay, it's irrelevant regarding the area under your curve, regarding your total impact. So we think that we have a measure that can not only capture the total impact of a paper, but can also predict it if you have only a few years into that. And prediction is an important word here, and I want to actually show you how this works out. So let's go back to the example where I started from, right? And we said this, we kind of left it there that the bad news was that we soon realized that we were actually in a very, very different league from the Genome Project, which of course really wasn't a surprise to us. We knew that, that the Genome Project was just about anywhere in the news you could hear and everybody was working on that. Look, but let me tell you that we could actually tell what happened to the Genome Project in the future. It turns out that the Genome Project paper follows the classical scenario how paper gather citations. This is what happens with them. The year 2, they reach their maximum, and then they slowly start decaying. And this is the analytical curve that our formula predicts that how should actually behave in the future. So up to 2003, the paper got 6,000 citations, I'm showing you there, and the formula is telling us its total impact is 13,000 citations. That you take time to infinity 100 years from now, if citations are still around, the total number of citations would be about 13,000, double of how much it got, and will slowly be decaying. So what happened to our paper? Well, it actually turned out that our paper did not follow very well this particular pattern. It did not follow in the sense that it did not saturate after year 2, and nor after year 5, and nor after year 7, and eventually it only started to peak out apparently at 2009 that it didn't really, and then this is somewhere we are. And if we fit the formula, it's not yet a perfect fit. We're waiting more years to pass and we'll know more actually, but the total impact actually ends up being about double. So this was, why is this different? Because the Genome Project is a classical paper. It was well anticipated and it was giving an answer that everybody knew it's coming, right? And therefore, the knowledge about it was built into the community, and everybody cited it and then slowly forgot about it, became common knowledge. However, the network science papers were building up their community as they go. Every time new people cited, new people were entering the field. So it was a very, very slow and delayed effect. The fundamental difference is really just the very, very delayed maximum time, and of course, a different fitness parameter that leads to a very different total impact. But this is really about individual papers. And individual papers are great, but how about the career of an individual? How does the impact of an individual change in time versus simply the citations of its individual papers? And again, we actually started out by looking at my own citation patterns to figure out how was really my career. So this is my first paper in 1989. And the red dots correspond how many citations each paper got after 10 years after their publication. Solid red means that we knew because already 10 years have passed on that. And then gray corresponds to those that we predicted using our formula because 10 years have not yet passed, but our formula was predictive about a year and a half ago. And then the empty circles correspond to those that we didn't even bother to predict because not enough time has passed to make an accurate prediction. Some of them we could already predict now. So if you look at it, you get actually a pretty depressive picture, right? So for the first 10 years, I was working a lot, publishing a lot, and not getting much done, right? Regarding the impact on the community. You see, I have lots of productivity, but not much really impact on the community. Then about 10 years later, somehow I did something that I started seeing the light, and that was the period of, things were really going well, papers were getting lots of citations. But the real question is not that. The real question is to what degree does my past performance predict what would be my future performance, right? What my dean and my chairman would like to know is that what will happen after that, or the hiring committee if I ever want to move from where I am right now. And the biggest question is that if you look at this curve, of course, not enough time has passed to figure out what has happened next, so am I really a dead wood at this stage, right? And this was really the question we posed, and the first insights we actually had is that I felt from my own example that once I published my first higher impact paper about 10 years after my first paper, then I somehow got it, and therefore if you see that after that I'm publishing one after the other, somewhat low impact but then some high impact papers as well. So clearly the patterns have changed. So the question we wanted to ask, when does it happen that you publish your highest impact paper? And that's easy to do. You simply go ahead and the most important, not the timing we cared about that, but I felt that once I published that paper, I got it. And from now on, I knew what to do right. What's the right mixture to write a good paper, and we were able to repeat it over and over. So that led to a very simple hypothesis. The hypothesis was that eventually, if you look at it, if you're publishing a high-impact paper, you should maybe see that coming by the citations increasing. But once you publish it, the average citations of the papers after that should be higher because you know the mixture and you got it at that moment. So we went out to test it. And the way we tested it is that we ended up identifying for a bunch of researchers their highest impact paper, mostly physicists in this case. And this is your N star, the highest impact paper. And we said, what is the impact of the paper? 2 papers before, 4 papers, and so on, as well as after. And we were hoping that there will be actually like an increase after you publish your highest impact paper, independent of when is that in your career. And to do that, actually, we ended up distinguishing scientists, physicists in 3 groups. Low impact, those whose highest impact paper is less than 20 papers, so that they haven't published any paper more than with 20 citations during their lifetime. Then average impact between 20 and 200, and high impact more than 200. So here's the depressing thing what happened. So this is actually the low impact researchers. There's not much impact, not much change in the citations at the higher impact level. But at the higher impact level, this is the medium, the average impact, and here is the high impact researchers. And what you see is, you don't see much, which is like, it's flat, then your highest impact paper, and then flat right after that. And we actually ended up calling in the lab this one the finger law. And hence that explains probably why I'm wearing this shirt today, just to help you remind you what is this. But what this actually telling us is that we can see impact coming, and no do we actually learn from it. You see, the citations are flat before that and flat right after that as well. So then, okay, fine, you don't see an effect before and after. When does that peak happen? Is that predictable? Is there a time when you're going to write your most impact work? And we can test that by simply looking, when do you publish your highest impact paper? As I said earlier, in my case, that turned out to be about not 10 years, but 13 years after I started publishing papers. So we took all the physicists and we said, what's the distribution of the time from your first to your highest impact paper? And this is what we got, you see. So this is the distribution of your maximum time, how many years. And if you look at it, this actually tells me that I am a dead wood, right? Because it says that, your highest impact paper most likely will come between 1 to 20 years between your career, and after that, your chances of publishing your highest impact paper drastically drops. And I'm somewhere here, so I'm done, right? So that's not good news for many of us in the first row, right? But actually, we started looking a bit more carefully. No, sorry, not about you. and we said, is this really just the impact of your work, or could this perhaps be explained by productivity? So we ended up taking each researchers, and we mixed up the impact of the papers they got. We kept the same time of the publication, but we randomly mixed the citations for each paper. And then we got a curve that is indistinguishable. What this is telling us? That this has nothing to do with when you publish your highest impact paper. It's telling us that this is simply, you publish your highest impact paper between 1st to 20 years in your career, because that's when you published most of your papers. You're pulling random numbers from a distribution, I'll tell you in a second, and that's where you're pulling most numbers. That's when you have the highest chance to actually publish your highest impact, and after 20 years, your productivity decreases, and with that, also the chance of pulling a very good high impact paper from the list. Furthermore, if we're going to simply rank from 1 to n your papers during your career, which we did for all of these guys, and we say, what is the impact in the order of 1 to 20, or 1 to n, then it turns out the impact to be completely flat, you see. So there is really no impact differences between the first or last or the middle papers. There is no peak there. So if I look in the space of your papers, your impact is completely uniform. So this means that your first or your last paper could be the highest impact. You never know. And there are actually evidence for that. If you go in the literature, you realize that, Frank Wilczek, who got the Nobel Prize in 2004, he got it for the very first paper he ever published, right? It took them many years to get it, but eventually it was here. And then we have examples of the opposite. So we have John Fenn, actually, a Yale researcher who after retired, actually not retired, I'm sorry, after forcefully was retired from Yale, he did the discovery that eventually led to the Nobel Prize that he got. And of course, a squirm of history is that after he was forced to retire from Yale, and he made the discovery, going to another university, Yale sued him for his lab books that realized that the ideas were already in his lab books and got all the patents back from him. So, but this guy actually really after retirement and publishing the work that he got the Nobel Prize. So the bottom line, there is always hope that your greatest discovery could be the first, the middle, or the very, very last paper you publish in your career. The impact is completely uniform in that sense. But there are actually certain patterns that are still, going over this random impact rule. And they're only apparent patterns, but some of them are actually real. And this is what I'm trying to show you here. So I'm showing the time-dependent productivity of researchers in function of time after their first publication. And again, the low, middle, and high-impact researchers. And you see that the low-impact researchers don't change much their productivity, but the very high-impact researchers do a lot. And there's actually, the impact is growing similarly, and there's not much in the low-impact category, but there's much more in the high-impact category. It turns out that this growth, the growth impact, is entirely and uniquely determined by the productivity growth only. So it's nothing about the quality growing, and we can show that. But the main thing what you see is that apparently productivity and impact improve during your career, and the improvement is very pronounced for those with higher impact and much less pronounced for those with lower impact. But the randomness that I showed you earlier, that your paper can be any time, your highest impact paper can be any time in your career, allows us to formulate a very, very simple model. And the very simple model says that if I want to find out what is the impact or the total number of citations your paper will get, for researchers, you are researcher I and paper alpha, how much you will get, it will be a product of a random luck element, because we all have to be a little bit lucky to actually publish the right paper, times a hidden parameter S that we're going to just call it for now a skill parameter, lacking a better word for it. So the hypothesis we have is really that when you publish a paper, you have an element of luck times an element of skill, and then they together determine whether your paper will be high or low impact. Very minimalistic model, right? It turns out that in order to test this model, you need to have three parameters. You need to figure out what is the luck element for each paper, what is the skill component, S component, as well as what is your productivity. And we ended up running this model across all the papers for about 100 years of physics literature, and finding out what are the correlations between these parameters by mapping this model into those publications. And we ended up finding this correlation function that tells you how the different parameters correlate to each other. And what I want actually, so once again, this is the luck element, this is your skill element, and this is your productivity. And what I want to emphasize here is that these elements in the matrix are zero. What does it mean they are zero? It means that the luck completely decouples from your skill, and as well luck completely decouples from your productivity. So really, the function that matters for you is a function that is only, there's a luck element that we all share, independent of where you are, who you are, what part of your career, we're all picking a luck element from the same distribution. And then we all of us actually have this skill and productivity element that is a separate story. And down the line, the most important is that this separation allows us to analytically derive what is this hidden parameter that can be uniquely assigned to each scientist, which for now we assume to be independent of time. So the assumption is that you have a skill component that independent of time tells you how well you can tune up your paper, how well you're actually going to use your ideas. And as I said, analytically we can calculate it, and it simply says that this skill component is the logarithm of the 10-year citations of your papers, summed over all your papers minus a constant that is the same for everyone. So the claim we're making is that from the citation patterns of a researcher, we can determine a unique parameter that for now we assume to be independent of time that tells you how well you're tweaking your ideas and turn into low or high impact publications. What does that mean? It means, let me just show you some examples. So I'm showing you researchers who we found from the data set to have low S. low hidden parameter. And you see, S is 1.49. The citations after 10 years after the papers were published are the best paper doesn't actually exceed about 20 or 25. A medium S researcher, however, has a couple of higher impact papers, and overall, actually, the citation patterns are higher. And the high impact researcher, every few years, ends up publishing a little bit of a blockbuster on their way. So this is if we assume that S is constant. Now, is it really constant? Is your skill doesn't change with time? So we ended up putting windows in the career of these individuals, and these are actually, we always look at careers who have already kind of people who start publishing already in the physics literature. And we measured for people roughly the same as how does actually the S change in time. And here's the remarkable fact. It doesn't. So this is the bunch of low S individuals, and you see their S is kind of constant in time if we use a rolling window. Here is the medium S individuals, and here is high S individuals. There's more fluctuations at high S because it's better result, but down the line they don't really change. So what we're kind of concluding from that is that really there is a parameter that each carries with them, and that parameter really tells how well we're taking this luck component that is actually for everyone available, and how well we're turning into project that eventually the community gets excited about that. And the most important thing about this one is that once you have S for a researcher, it's very predictive of its future impact. So how do we measure future impact? We say, how does the total number of citations of your papers change with time? And here we are. If you take an S 1.2 researchers or 5.3, we can actually analytically derive the total number of citations you would get. That would be the red curve during the career in terms of the number of papers you published. And here's the real data. And I can show you hundreds of curves like that, and it's terribly accurate. That is, that parameter tells you how would your total number of citations change in time, despite the fact that you keep publishing more papers. It will also tell you how the age index will change in time. And this is what I actually want to emphasize, that the traditional measures we use, the citations and age index, are time-dependent measures. Hence, it's impossible to compare different individuals at different stages of their career. It's impossible to compare individuals in different fields and so on, because they have different citation patterns. What we're finding is that this S parameter, this hidden parameter, is at the bottom of all of them. Once I have it for a researcher, it deterministically almost determines how this is going to develop in time in function of the productivity, a number of papers they published. And the last example I would like to show is that this also allows us to rank researchers, and we ended up ranking physicists based on the S parameter and a bunch of other things like age index, highest impact paper, total number of citations, and productivity. And what we are finding that S is the best predictor whether someone will actually get the Nobel Prize, and the worst predictor is productivity. So down the line, and kind of the total number of citations and the age index and the highest number of citations are comparable to each other, but they're weaker than S. So clearly, S is capturing something intrinsic about what we do. So we have at the end, we have a very simple model telling us that the impact of the paper is a luck element that we all share. We all pull the same luck points from everywhere, times a skill that is unique to all of us. And the way we visualize this works actually is that each scientist simply selects a project that is a luck element, and then it improves it with a factor of S. And this means that high impact publications come from people with high S who were also lucky to pick a high luck element. And everyone can publish a low impact paper independent of S by simply picking a very low luck element, right, a low P. And the reason why we think S is important is because it does not vary during a career, and it predicts the citation dynamics, it predicts the age index, and it's also the best predictor of exceptional achievement like Nobel Prizes, Boltzmann, and other physics prizes at least. So that's about the career of an individual. Now, the last thing I want to say is that we live in a society where we must share credit. What do I mean by sharing credit? Well, there were some individuals in science where the credit was obvious where it belongs to. Think about Einstein's paper, had one author of relativity. It was pretty clear that he gets all the credit. So did Darwin. But we don't live anymore in those areas. And then it becomes really fuzzy to determine where the credit goes, who should get credit for a particular work. And let me show you 3 examples. So these are 3 papers. Each of them got a physics Nobel Prize. would you be able to guess whether it's the first, the last, or the middle author who gets the Nobel Prize? Well, in the first paper, it was the last author who got the Nobel Prize. In the second, it was the first author to get the Nobel Prize, and in the last one was the middle author to get the Nobel Prize for those papers. So how about here? Could you figure out who will get the Nobel Prize? Well, it's obvious, right? It's in there. So the question is, how does this work? We publish these papers with hundreds of co-authors, yet the community comes along and picks some from those and say, you get the credit for all of this work, and for the rest is forgotten to a certain degree. Or is it really forgotten? How much credit is really assigned? And I would claim that this is a collective process that we can figure out, because the community collectively knows how to assign credit. That's how we assign prizes and so on. And we can figure it out by the co-citation patterns. And let me show you the idea what is behind that. Well, take this paper who got the 2010 Nobel Prize in chemistry. The paper for which the Nobel Prize was awarded was published in 1976 by 2 authors, but yet only the second author got the Nobel Prize. And the reason is very simple. When the community looked at the subsequent papers, the most cited papers that are co-cited with the Nobel Prize winning paper, they all turned out to have the second author as a co-author, and the first author disappeared from the picture. So the second author continued to work on this problem and established himself as the person who made the discovery, and therefore the credit to the first one disappeared. Well, how about this other one, so that's where the Nobel Prize goes. How about here? Noselov and Game, many of you remember, this is the Graphene Nobel Prize. And when you look at the most co-cited papers with the Nobel Prize winning paper, they're all joined by both authors. Hence, not surprisingly, we can't really distinguish between them, and they both get the Nobel Prize. So my claim is that this is how we do it. We look at the career of individual and say, is this line of work really comes out from the career of that individual? Does he or she continues to work on that, establish himself as their own area? And that's how we actually decide who should get the credit. Not because we don't know who really made the discovery, right? We have no way of figuring that out, but we have figuring out the career of an individual. And it turns out that we can quantify that. Based on this idea, you can write a very precise algorithm that the visitor in my lab did, Wei Shen. And that allows us to assign to each author in a paper a credit share. What is the credit share? It captures, based on the co-cited papers by the co-authors, how well the community assigns credit. The idea is that if you publish a paper with several authors and I keep citing together that paper with lots of your papers and I ignore your co-author's paper, I'm telling that that line of research that I'm citing is really your paper. And if we do that and apply to that, it tells us that in this particular paper, the credit share for the first author is 0.22, the second time is 0.72. So the community views that 72% of the credit goes to the last author and 22% to the first one, and therefore it's his paper, and that's where the Nobel Prize goes. In the second case, it's half and half, exactly split between the two. So we ended up applying this one to all Nobel Prize winning papers. Why did we do that? Because in the case of Nobel Prize winning papers, already the credit has been assigned by the community and it's publicly known. And we find out that in the red cases where we get it right, the black cases where actually we make a mistake, in a very small fraction of the cases we make mistakes, and each of these cases is fascinating wherever we make a mistake. There's a story behind that, why are we making a mistake? And let me show you one example where there's a beautiful story behind that, and that's an example that everyone knows, at least principle, the Nobel Prize for this was the Higgs boson. And some of you may remember that the Higgs boson was when it was obvious that there would be a Nobel Prize to it, but it wasn't clear who should get it. Because there were 3 papers within 1 year that all published the same result. There was a paper by Engelert and Braut, then a few months later was the Higgs paper, and then a few months later was the Kibble and his collaborators, where they ended up publishing just about everything that the first 2 papers did, plus more. And so this actually shows that I can go ahead now and assign credit not only for people within a particular paper, but also people who did not publish together, but they're in the same community, because the same question applies. Where do we assign the credit between people in the same community as we co-cite them? And the answer is very simple. You see, Higgs gets about 40% of the credit. So that's right. Not surprisingly, he's the one who is the face of the brand. But the second most cited, in function of the time, the second most credit really goes to Kibble. And where does the Nobel Prize go? he goes to the first, and he goes to anger because Braut passed away. So this is a really funny case where we actually think that the Nobel Prize committee did not follow what the community thinks about that one, because the community has clearly voted that Kibble is the second most important person. So therefore, the prize was given to these two gentlemen, and not surprisingly, Kibble is not particularly happy about that. and even the Nobel Prize winner, actually, Hicks says that I rather hoped before the prize was announced that that Kibble will actually get the prize. So even Hicks himself has announced. So that takes me to my last slide. What did I try to tell you? Well, what I showed you, the first thing is that we can't really see when success is coming, nor do we actually learn from it. Somehow it ripples through our career without having obvious signs when it's going to come. But the productivity and impact do improve during for high-impact researchers, but fundamentally behind that improvement is a random impact rule, that you never know when your hit comes. And the most important thing is that we were actually able to formalize how success happens in science by saying it's an element of luck and skill. And surprisingly, we find that skill doesn't change much during the career of an individual. Luck does. And the combination of two gives us actually a way of really predicting very accurately the impact of individuals. And I'm just going to show the acknowledgement slides for my lab. And thank you very much for your attention.
Great. Thank you. Obviously, something all of us need to think about deeply. We have time for a couple of questions, but I'm going to start off. So how does this cross with institution?
That's a very good question. So, I mean, that's a variable that we have not explored in this context. We explored in a different context of how people move. And I think that there are lots of interesting questions. And the interesting question really would come, is your skill element determined by your educational pattern, and to what degree? That's where it would come from, because already the skill element has been already very separated. What I would expect that there will be institutional and other correlations how you built up that skill parameter. But once you build it up, it's a little bit independent of where the individual seems to be.
Okay. Maybe one other good question? Sir, you had your hand up first. Yep.
The citation patterns you mentioned seem to me similar to tweets or something on social media going viral. Mm-hmm. So I was wondering if you considered the parallel aspect
yes,
of that.
We actually think that it applies to that. There are lots of similarities, and we had a project where we tried to kind of achieve that. For various personal reasons, it didn't actually lead anywhere, but we think still convinced that the same ideas can be applied. The formula has to be tweaked because there are some differences between but what we think that there is a skill element, and we think that the skill element has to do with the fact of whether the tweets you write, the content and the way you formulate it really gets you above the others. So to eliminate from that actually how many followers you have. So it's independent of the follower, should be independent of the follower aspect, should be your skill of writing the tweets. So we try to deconvolute it, but then the student had to leave to a different project, so we couldn't finish up. We hope to return to it.
Good. Okay. Thank you. Great talk. We should move on to the next. Perfect. Thank you very much. Oh. I'm supposed to give you this gift, too. So let me do that. Thank you very much.
Oh, sorry.
And you get to get it. Oh, yeah. Yes. Great talk. So the next talk is by Nicholas Christakis, formerly of Harvard, now of Yale, where he directs the Human Nature Lab. Nicholas is famous for his best-selling book, Connected, and of course for all the great work he's done over the years. And he brings to the notion of computational social science really a strong background in medicine and genomics, which I think is a great thing to have here. So Nicholas, without much more, please.
Thank you very much. Can you hear me in the back all right with this microphone? Yeah? Oh, we're turning it on. That's terrific. Thank you for the introduction. Most of the work I'm going to present to you today is done jointly with James Fowler at UCSD. And I'd like to sort of add an element to the conversation we've been having the last couple of days, and I hope we'll continue to have. And that is that I think that biology is a very important constraint on computational social science, because it places constraints on and it shapes modern technologies that we use to communicate and that we actually use to generate the kinds of data that most of us also study. In fact, our biology shapes our behaviors and is, in this sense, a foundation upon which all else is built. Because after all, human beings have been embedded in social networks for tens of thousands of years, and not just since the 1990s when we developed the kinds of technologies that we've been using to collect the kinds of data that we're interested in. And I actually see social networks as a kind of biosocial phenomena. Networks have biological predicates and biological antecedents, and they have biological consequences. I think the structure and function of human social networks are deeply rooted in our evolutionary past. Think about this. Human beings are unusual as a species, although we share this with certain primates, elephants, and dolphins, in that we form stable, long-term, non-reproductive unions to other members of our species. Namely, we have friends. Why? Why do we do that? That's a very specific and unusual behavior that's uncommon in the animal kingdom, and yet we take it for granted and we do it every day and all the time. And in addition, and this is in fact one of the problems that James and I put before us, and in addition, it's been observed for a long time, since the ancient Greeks in fact, that we prefer the company of people we resemble. We actually, as a species, evince homophily. Why do we do that? Why do we prefer the company of people we resemble? And actually, the contemporary accounts for this primarily say, well, you prefer the company of people you resemble because you're more comfortable in their presence. Why are you more comfortable in their presence? Because they resemble you. It's a kind of tautological explanation for homophily, but we are hoping to provide a deeper explanation, not only for why do people have friends, but also for why we prefer the company of people that we resemble. Now, like other species, human beings can be seen as evolving under the pressure of the environment that they face. And this environment may be seen to be composed of three types. There's the physical environment that our species faces, the sunlight, temperature, water supply, and so forth. There's the biological environment, predators or prey or pathogens, but also there's the social environment, sort of who and what is around us. And this social environment has typically been neglected as a force of natural selection. And yet social factors can obviously be crucial, and in fact they may have led us to being intelligent. In fact, the social brain hypothesis posits that one of the reasons we are intelligent is the necessity not to make tools, but rather to make friends. And our assembly into groups may have accelerated, if not created, the tendency to think that we in turn bring to bear on all the kinds of problems that all the smart people in the room are doing. But what's interesting about the social environment, more so than the physical and biological environment, at least until very recently, is that we humans, both individually and collectively, create the social environment. So we create this environment, which then imposes selection pressures on us. Those selection pressures feed back and reshape who we are, reinforcing then the further creation of the similar environment in the future. And you can get this vast feedback loop across evolutionary time that may be accelerating our tendency to be social in very specific ways. And in fact, as a further matter, we may actually be creating these social pressures over shorter time horizons and not just over long time horizons. So here's an image of a natural social network, which is the kind of social environment we create and then proceed to live out our lives within, no different than other kinds of environment, I would argue. Each of us chooses our friends, inherits our relatives, chooses our colleagues. All of those people are doing the same thing, and we proceed to create this ornate structure, this network, and live out our lives in a specific location within this structure. And as a result, these structures proceed to affect us in a variety of ways. This is taken from one of our first studies, and it helped us to understand the role of social networks in obesity. Every dot is a person. Every line between them represents a relationship between two people. The dot size is proportional to people's body mass index. Bigger dots are bigger people. And we also color the dots yellow if people are properly obese. And if you look at this image, you might be able to see clusters of obese and non-obese individuals within the network, little outbreaks of obesity, perhaps. Nevertheless, the complexity is still very high, visually speaking, and even if you grant me that you can see the clusters, it's not really obvious how much clustering there is, or is there more clustering than due to chance, or how big the clusters are, or most importantly, what might cause the clusters. So in that paper, we did a little computations of various kinds. One of the computations sought to establish that these clusters in the fabric of humanity, in the network, exceeded chance, and to measure the diameter or the radius of those clusters. And this slide shows on the y-axis the increase in the probability that a person is obese, given that a social contact of theirs is obese, and on the x-axis the degrees of separation between the two people. And it just says that if your friends at 1 degree of separation are obese, you've got a 45% higher likelihood of being obese yourself. And if your friends at 2 degrees of separation are obese, you've got about a 20% or 25% higher likelihood of being obese. And if your friends at 3 degrees of separation are obese, you've got a 10% higher likelihood of being obese. It's only when I get to 4 degrees of separation, to your friends, that there's no longer a relationship between that person's body size and your body size. So if I knew nothing else about you, and the body size of this individual who's almost certainly a stranger to you, your friend's friend, for example, I would be better than chance able to predict your body size. So this analysis just tells us 2 things. First, it tells us that there's more clustering in this trait, this obesity trait, than due to chance. If it were chance, it would be 0% across the board. There'd be no excess risk as you moved in geodesic space. And second, it tells us that the radius of the clusters is to about 3 degrees of separation. But it doesn't tell us the cause of the clusters. And there are at least 3 possible explanations for why these clusters might arise. There's an idea of a social domino effect or induction or a kind of social contagion whereby your weight gain causes my weight gain and my weight gain causes your weight gain in a kind of spreading process in the network. Another possibility is birds of a feather flock together, or love of like, or homophily. So here the idea is that I preferentially form an attachment to Laszlo because he and I have a similar body size, for example. Not that his body size, alas, contributes to my body size. And the last possibility is that actually it's neither that my body size causes his body size nor that he and I form an attachment because we have a similar body size, but rather that something in the environment jointly contributes to his and my body size being similar. And all 3 of these processes are typically present in any social phenomenon, and a variety of mechanisms might underlie each of them. And we have used a variety of observational and experimental strategies to sort this out in a broad set of circumstances, and we found evidence that weight gain and other phenomena spread to about 3 degrees of separation in a kind of social contagion. And in the process, we also documented homophily on a variety of traits, confirming not only the existence of social contagion, but also the widely and long recognized phenomenon of homophily, which was appreciated beginning in the 1850s, has long been understood as a possible confounder for when you find individuals with similar traits. Now, eventually we began to become interested in other sorts of phenomena with respect to contagion and homophily. So for instance, we began to look at human emotions like anger or happiness or fear or disgust. Now, when human beings have emotions, we show them. Why? Why do we show our emotions? It's not hard to construct an argument from evolutionary biology as to why it would increase our fitness to experience emotions, but not only do I experience an emotion, but I show it. And not only do I show it, but you can read it. And not only do you read it, but you copy it. It would seem that emotional contagion is a very deep and fundamental aspect of human emotional experience. And this function of emotions suggests that whatever other advantages they offer, they're also a primitive form of communication. Now, we're accustomed to thinking about emotional contagion as fleeting and involving just a pair of people. So a few years ago, when I presented some of this work in New York City, I said, you know when you're in the subway and the person across the car from you smiles at you and you just instinctively smile back? And they said, we don't do that in New York City. And I said, well, everywhere else in the world, that's normal human behavior. When someone smiles at you, you just smile back. And so we're accustomed to thinking about emotional contagion as fleeting and involving just a pair of people. But of course, there can be broader collective expressions of emotions, such as anger in the form of riots. And we wondered whether emotions might spread beyond pairs of people, and might they do so in a more sustained way than riots? Could there be a kind of a quiet riot? Could there be a kind of below-the-surface emotional stampede rippling in these social networks? Maybe emotions spread across ties well beyond pairs and in a more sustained fashion all the time. In fact, maybe emotions have a collective existence, not just an individual existence. So we made what I think is the first network map of human emotions. Here every dot is a person again. Lines represent different sorts of social relationships. The yellow dots are happy people. The blue dots are sad people. The green dots are in between. And if you look at this image, once again, you can see clusters of yellow and blue dots. These clusters also spread to about 3 degrees of separation. And there are other structural information that's visible in this graph, including that the unhappy people tend to be more likely to be on the periphery of the network and not just in the middle. And similar analyses to those that we've done earlier for other sorts of things persuaded us about the importance both of contagion, emotional contagion, and of emotional homophily in this graph. But to get around some of the problems that we've been facing using observational data, we eventually began to do some experiments. This is a paper we published now in 2010, so a few years ago. In this experiment, subjects were brought into the laboratory. They were strangers to each other. They were randomly assigned into groups of 4, and they were given a little bit of money. And if they contributed the money to this collective group of 4 that we had created, the investigators doubled the money and then divided it amongst the 4 individuals in a kind of public goods game. And then a bell would ring and they'd be randomly assigned to new people. And then a bell would ring and then be randomly assigned to new people for up to 6 time steps across time. And so what we were able to do with these data is to create and then reconstruct artificial social networks and test the idea not if I'm kind to Santo, does he reciprocate the kindness and is he kind to me, but rather could we define evidence for a kind of pay it forward phenomenon, whereby if I'm kind to Santo, Santo's kind to Alex, Alex is kind to Laszlo, and there's a kind of spreading process in the network, here shown experimentally. And that's exactly what we were able to find. What we were able to show is that if Eleni is kind to Lucas in period 1, Lucas is kind to Erica in period 2, Erica is kind to Jay in period 3, and Jay is kind to Brecken in period 4. And this is one of the more bizarre results to come out of my lab in the last few years. Because what I've just told you is that how Jay treats Brecken depends on how Eleni treats Lucas, even though neither Jay nor Brecken ever interacted with or saw Eleni or Lucas. How you 2 guys treat each other depends on how those 2 people treat each other, even though you don't know who those individuals are. A kind of spreading process in the network, here not observed observationally, but rather shown experimentally. So Michael Macy's experiments also showed something similar as he presented yesterday. And in fact, in addition, this geodesic spread of kindness is different than the temporal persistence of kindness highlighted in yellow, such that if Eleni is kind to Lucas, he learns to be kind, and with every subsequent interactions with other people, he is kind with those individuals as well. In fact, if you fold back all the downstream kindness, for every extra dollar that Eleni gives Lucas, the network functions as a kind of matching grant, doubling the benefits that accrue to the population as a result of the individual's actions. So one of the ideas that we've been converging on with respect to human social networks is that networks function as a kind of magnifying glass. They magnify whatever they are seeded with, good or bad. And in fact, a deep reason we might be predisposed to live in networks, and not just in groups, which are amorphous, is that the network might permit and foster cooperation and enhance the returns on cooperation, and that the structure of the network may be highly relevant to this successful behavior and, in fact, to other behaviors, as I'll talk about in just a moment, as both this experiment and several other subsequent experiments we've done have shown. So network structure is highly relevant to all kinds of phenomena like cooperation, but also the expression of immunity, the spread of germs, the spread of information, and social learning. These very fundamental and deep aspects of our humanity and our survival are intrinsically connected to the fact that we make networks with particular topologies and particular properties. Now, this work with emotion and with cooperation, which are so fundamental, coupled with the idea that networks have a function, got us to thinking about the evolutionary basis of networks themselves. Now, human social networks, wherever they've been mapped, always look strikingly similar. In the upper left, you have about 56 high school students and their friends. On the upper right, 105 college students and their friends. In the lower right, you have telephone data taken from Laszlo's lab, 400 people out of 8 million individuals and who they're calling. In the left, you have a scientific co-authorship network. Visually and mathematically, these networks look incredibly similar. But they never look like this. We never go out into the world and map a network of human beings and find that the network naturally looks like this regular lattice or anything remotely like it. Why not? Why don't we form these kinds of networks as opposed to some other kind of network? Well, the striking pattern of human social networks, their ubiquity and their apparent purpose, beg the question of whether we evolved to have networks and to have particular kinds of networks in the first place. So now the questions become, why do we form networks in the first place, and why do they have the structure that they do? And to understand this, we need to dissect network structure a little bit first. So first of all, notice that in this type of a network, every position is isomorphic to every other position. Everyone has 8 friends, every one of their friends has 8 friends. If you took this surface and wrapped it around the surface of a sphere, or more properly in 3 dimensions, the surface of a torus, every position would be equally distant from the periphery as every other position. But of course, as most of us know, that's not what real networks look like. Real networks have heterogeneity in position and location within the graphs. Now, most of you will know what I'm about to say, but I'm going to quickly introduce it for those of you who do not. There are a number of ways you can quantify people's positions in networks. Let's just quickly look at 3. First of all, look at node B in the upper left and node D. B has 4 connections and D has 6 connections. This is known as the degree of a node, and people generally know this about themselves. You have 10 friends. You have 8 friends. I have no friends. People know this about themselves. But now look at nodes C and D. C and D both have 6 connections, but of course we can see that C and D are rather different. And I can cultivate this intuition in you by asking you, who would you rather be if a deadly germ was spreading through the network? You'd rather be D. Conversely, who would you rather be if a juicy piece of gossip were spreading through the network? You'd rather be C. And this, of course, is the centrality of a node and can be quantified in many ways that we've been discussing and most of you know about. And finally, look at nodes A and B. A and B both have 4 connections, but the difference between the two is that A's friends are friends with each other. There's triadic closure or transitivity, whereas B's friends are not friends with each other. They're reach further into the network. So the friend of A's is back again a friend of A's, whereas the friend of B's is not a friend of B's. And if you were a prehistoric human and I asked you, who would you rather be if the task was to go out and work together to kill a mastodon? You'd probably rather be A than B. Work well together with your friends and go kill the damn thing. But now if the challenge was not to kill the mastodon, but to find the mastodon, you'd rather be B, because B can reach further into the graph to get novel information, for example, about the location of predators or prey. So in one of our first papers about this, now also published about 5 years ago, I can't see the date from here, we used a simple twin study design to quantify the extent to which these 3 traits were heritable. And on the y-axis, we show the heritability of the traits, and on the x-axis, the 3 traits. And what we found was that 46% of the variation in how many friends people have can be explained by their genes. Now, this is not a very shocking result. Most of us know that some people are born shy, and some people are born gregarious. People vary in their taste for friendship. Not all nodes in a network are the same. Nodes have heterogeneity. But in addition, we found that some of the higher order properties also can be heritable. So 47% of the variation in their transitivity can also be explained by your genes. This is another very bizarre result to come out of my lab in the last few years. Because what I've just told you is that if you have Tom, Dick, and Harry in a room, whether Dick is friends with Harry depends not just on Dick's genes or on Harry's genes, but on Tom's genes. Whether these 2 guys are friends with each other depends on this person's genes. How can that be? Well, we think the reason is that people vary in their tendency to introduce their friends to each other. Some of us knit the networks around us together and foster transitivity, and others of us do not. And finally, even 29% of the variation in how central you are in the network can also be explained by your genes. So where you are in this vast fabric of humanity depends in part on your genes. And the fundamental reality of our desire for particular kinds of connection, it turns out, has always been with us. And it turns out that this variation in topological preferences, which I just discussed, and the exercise of choice in friendship selection can result in, or actually reflect, a genetic structuring of the human population. So what this means is that each of us lives and dies not only as a function of our own genes, but also as a function of the genes of the other people around us, who are people we chose. Each of us lives in the sea of genes of others in the network, people that we specifically, little niches that we create around us. And in a subsequent study, we found evidence for genetic niches within networks, here shown with respect to the alleles of one gene, DRD2, where there's a correlation above that expected due to background rates in variants of the gene seen in groups of interconnected individuals. Now, this cannot be due to social influence. My genes aren't causing your genes. If I find a similarity in the allelic variants, the variants of the kinds of gene I have, between me and you, my friend, it must be because I chose you. It must be because of homophily. It can't be because, I've caused your genes to flip and become just like my genes. And actually, if you look at the allelic variants, the types of a particular gene in this population, you find clustering by the variants as shown by the colors and the legend shown on the right. So there isn't just phenotypic homophily between individuals. It's not just that smart people hang out with smart people and tall people with tall people and so forth and so on. There's genotypic homophily as well. And in this initial paper, we showed that it drills all the way down to the single nucleotide polymorphism level. So it's not just that you have some genes that predispose you to height and your friend has other genes, nor even is it that you have a specific gene and then your friend has that same specific gene. Down to the single locus within your genome, the variant you have is very likely to match your friend's variant above background rates and more than due to chance. And this structure can arise for several possible reasons. Now, one reason is just something known in genetics as population stratification. So here the idea is that there's a kind of ethnic stratification of the population. If I'm Greek and I preferentially hang out with Greeks, it shouldn't be surprising that my friends will have genes that are similar to mine. We're both Greek, right? That's the individuals that I'm hanging out with. So that's obvious and trivial and needs to be controlled for if you're going to begin to look at some other factors. Because another possibility is that above and beyond that, there could be choice of friends. So I go out into the world and I'm picking who my friends should be. Maybe I'm selectively picking individuals who resemble me on a genetic level. A third possibility is not that I'm picking individuals, but rather that I'm picking an environment. Let's say I'm very good at running, and I go to a place where I can exercise this trait of running. And in that place, I find other people who are also good runners, and that's the people from whom I pick my friends. So that might explain it. And a third possibility is that selection by others. So here, for example, Santo picks in smart people to come to this meeting, and here we find other people who we befriend, and so now we have similar genes, but because of Santo's agency in selecting individuals to come to this meeting. And so, what happens to us may thus depend not only on our own genes, but the genes of our friends. And there was a beautiful paper published by another group a few years ago in PNAS that showed, no, actually it wasn't in PNAS, in another journal, that showed this in hens. And what they found was that the feather condition of hens, feather condition is a very important phenotype if you're a hen, the feather condition of hens depended not as much on the genes of the index hen, but rather on the genes of the hens that were experimentally caged around that index hen. So they created networks of hens, and they looked at the phenotype of the hen and saw how that phenotype depended on the genotypes of the nearby hens and found a relationship. So this means that there may be genetic origins and genetic implications of friendship. And this in turn means that natural selection might operate at the level of friendship unions and not just reproductive unions. So natural selection may work not just in who we choose to mate with, but who we choose to befriend as well, because those individuals often confer fitness advantages upon us as well. An organism's fitness may depend in part on the friends it chooses. And in fact, we may be metagenomic. Now, this idea of metagenomics has become very popular in the last 10 years with respect to bacteria. So we look at groups of bacteria in a packet of dirt, or we think about human beings. Your fitness depends not only on your own genes, but the genes, now it's increasingly accepted, of the bacteria in your body. But why should your fate only depend on your own genes or the genes of the bacteria in your body, but not also on your genes plus the genes of your friends, right? So you can begin now to expand our vision of what counts in terms of determining our fitness. Now, the people to whom we are connected provide all kinds of important capabilities, from the ability to reciprocate cooperative exchanges. If you're kind to people, you would want preferentially to pick people who are also prone to kindness. You would want homophily on cooperativity, for example, to enhance the rate of return on this investment. And the people to whom you're connected also provide other advantages. For example, the ability to ward off infections, or the ability to transmit or exploit useful information. And consequently, the fitness advantages of some gene variants might be influenced by their parallel presence or absence in other individuals to whom a person is connected. So if you're the first person on the planet to just dumb luck evolve the capacity for speech, the fitness advantage to you for that, this is a toy example, the fitness advantage of you to that is 0 unless you can find another person to talk to. And then the moment you can connect to someone else to talk to, the advantage to that mutation suddenly rises dramatically. So homophily, for example, on the capacity for speech, as in the capacity for cooperation, and as in the capacity for certain immune functions, might depend on the genes of other people to whom we are connected. And in some theoretical work that we did that we published a couple of years ago in partnership with Martin Nowak's lab, we showed that natural selection can give rise to homophily when individuals engage in social interaction in a population with multiple observable phenotypes. Here, the payoffs to interactions depend on whether or not individuals have the same or different phenotypes, and individuals have a preference that determines how likely they are to interact with others of their own phenotype, homophily, or the opposite phenotype, heterophily. And what these models, the bottom line is what these models suggested is that homophily tends to evolve under a much wider range of conditions than heterophily, even when there's a fitness advantage to heterophily. Now, the intuition here is the following. In a population where everyone is the same type, if you derive an advantage from connecting to someone of the same type, it's very easy to find a person of the same type, because everyone's the same type. But in a population of heterogeneous types, if you derive an advantage from connecting to someone who's of the opposite type, there are fewer such people and it's harder to find them. So paradoxically, you can actually get homophily evolving in this model, even if it's more advantageous to be heterophilous than it is to be homophilous. Now, we tested this idea in another very recent paper published just a year or so ago in PNAS. In this paper, what we did is we looked at a large sample of people, and we looked across all friendship types. We looked at large groups of friends, and we had 500,000 SNPs, or we were able to, these people were genotyped at a very fine level, and we explored whether these ideas that I've been presenting are present across the entire genome. And we compared the genotypic resemblance, here quantified as the kinship coefficient, or the probability that two alleles sampled at random from two individuals are identical by state, between pairs of friends and pairs of strangers drawn from the same population. So what we did is we looked at every pair of individuals in this population, and we said, well, how related, and these were unrelated individuals. They were not biologically related, they were friends. And we said, well, how related were they as if they were actually kin? And we found that on average, people resembled their friends as if their friends were at the fourth cousin level. They were not their fourth cousins. We knew this because we had these genotypes, but the people that they were befriending looked like approximately fourth cousins to them. And so the kinship amongst the friendship pairs was shifted to the right compared to the stranger pairs in the same population. When we looked at individuals who were strangers to each other, there was no relationship among those individuals. So what we were able to do is show that friends are more genetically alike across the whole genome, and not just over the specific SNPs for DRD2 and some other genes that I showed you earlier. Friends exhibit significantly more homophily, positive correlation, than strangers in genome-wide measures. Compared to strangers, strictly unrelated friends who are not cryptic kin had higher kinship coefficients. And indeed, on average, friends had a kinship coefficient that corresponded roughly to the relatedness of fourth cousins. And in other results, we also found that in some regions of the genome evince heterophily, or this opposites attract feature. On average, across the whole genome, it's homophily, but certain regions of the genome, we're now beginning to find some interesting ideas about why opposites might attract. Here, not sexually, but in terms of friendship unions. Now, we expect that there are likely to be dozens and maybe even hundreds of genetic pathways that form the basis of correlation in specific genotypes. And our sample gives us enough power to detect just a few of these pathways. So we look across the genome, we see, well, what kinds of traits do we have evidence, preliminary evidence at this stage in the work for homophily? We find that actually people tend to befriend other people with similar senses of smell. It's not that they smell good to you, although they might. It's that they and you and your friends tend to have similar taste in olfaction, similar senses of smell. We also find homophily on linoleic acid metabolism, and in a prediction we had made a few years earlier, we found heterophily in the immune system. People preferentially attach to individuals with dissimilar immune systems, which makes sense theoretically, because if you're immune to one pathogen that's spreading in a network, you'd rather surround yourself by individuals who are immune to opposite pathogens. Because you're immune to A, actually surround yourself with people who are immune to B. B doesn't get you because it can't get through your friends. A gets through your friends, but you're immune to A. So we find heterophily in immune genomes in this individuals, in this sample. And in fact, we go on to explore another idea, If homophily is generally contributing to evolutionary fitness across a wide variety of traits, we would expect to see signs of positive selection for genes that exhibit positive correlation between friends. And so here the idea is, let's look at the whole genome now, and let's look at those segments of the genome, wherever they occur physically, those genes, those SNPs, those single nucleotide polymorphisms, let's look at those where across our sample, people are most like their friends. And then let's look at the next most likely, and the next most likely, and the least likely of their friends. And now let's test in which of those groups of genes is there most evidence for recent natural selection. Because if it's the case that homophily is advantageous in the ways I was alluding to earlier, there should be natural selection, there should be positive selection for those specific loci in the genome. And that's exactly what we find. Here on the y-axis is positive selection measured by Partis-Sabeti's composite of multiple signal scale. On the x-axis is the genotypic correlation. And we find for the quintile of genes that are the most homophilous, the friends have the highest between friends, that's where you have the highest evidence of recent selection. And in the control group of strangers, you find no difference across the categories. So other people around us have not only reproductive value, but also associative value. And in fact, we believe that one of the things that may be happening is that this reflects the extended workings this suggests that that our capacity to make friends with unrelated strangers may have played a role in human evolution. Humans may, when choosing friends from among individuals who are strictly not related to them, come to choose individuals that, after all, resemble them on a genotypic level. And this may possibly reflect the extended workings of a kin detection system, which has long been postulated in humans, but difficult to show, whereby our friends are a kind of functional kin. We go out, we're looking for our kin, we don't find our real kin, but we pick the next best thing, which is our people who resemble us on a genotypic level. So the people around us have not only reproductive value, but also associative value. And it's important to sort all this out because genes might be associated both with network topology and with behavioral outcomes. And these interpersonal effects might depend upon both. So for example, other labs around the world look at how genes, for example, in the dopamine and serotonin pathway, affect different sorts of behaviors, obesity, happiness, addiction, and altruism. Earlier, James and I and others have done work showing that social networks can affect these behaviors. Now I've just shown that genes might affect these social networks. And what this suggests is that a lot of the models of behavior genetics that look at how genes affect behaviors may be partially misspecified because they're avoiding or ignoring this backdoor path. Maybe the way genes make you happy is not by what they do inside your brain and inside your body, but what they do outside your brain and body. Maybe the genes affect how many friends you have or which friends you pick and create this backdoor path, and ignoring this path means you over-assign rationale and import to the top path, ignoring the bottom. Now I'm going to present results from one more paper, and then I'll close with a couple of ideas. If social network structure has biological and genetic antecedents and evolutionary significance, it begs the question of how ancient this structure is and whether we can document such structural features in non-modernized populations. So here the idea was, if networks are rooted in our evolutionary past, if we could go back 10,000 years and study us 10,000 years ago, the networks maybe should look just like they do today. Now, obviously, we can't do that. So instead, we decided to map the social networks of the Hadza hunter-gatherers in a paper that was published a couple of years ago. So we partnered with some of the world's leading ethnographers of the Hadza. The Hadza are one of the last hunter-gatherer populations on the planet. There are only about 1,000 of them left. They live like we did during the Pleistocene. They have no material possessions to speak of. They hunt and they gather for their food. They sleep out under the stars. And we made a photographic census of all adult living Hadza, a kind of Hadza Facebook. And my postdoc, Corinne Epicella, went into the field, drove around for 3 months, and every Hadza she found, she got them to identify who their friends were. And when we mapped the networks of the Hadza and studied them, we found that with every kind of quantitative parameter that's used to study modernized networks using telecommunications, there was no difference between the Hadza networks and our networks. The degree distribution was the same, homophily was the same, geographic decay, transitivity, degree assortativity, and even reciprocity. And so many of these features actually have functional significance in human populations. And actually, social networks are very deeply rooted in our evolutionary heritage. And here's a graph of the network, and now we also highlight who's a cooperative individual and who's not. And in ways I won't go into today, we find evidence that the cooperators are clustered within the graph compared to the non-cooperators, and this network mathematically and visually is indistinguishable from the kinds of networks we make, despite the fact that in the intervening 10,000 years, we've invented agriculture, we've invented cities, and we've invented telephony. Now I'm going to allude to one more experiment and then I'll close. We then, using computational social science techniques, tested some of the ideas we had in the Hadza by doing some experiments using Amazon Mechanical Turk in ways that Duncan was highlighting yesterday and that Michael has also exploited. So here we recruit thousands of subjects to come and live in these temporary virtual societies that we make. And one of the promises of computational social science, in my view, is that it gives us the opportunity for the kinds of experiments that Duncan and Laszlo and others have been discussing, and Michael. So for example, in this experiment, and I won't go into the details, what we were able to do is we were able to replicate the Hadza preferential attachment to cooperators in a dynamic setting by creating these temporary virtual societies, recruiting individuals, and showing that under certain topological regimes, we were able to preserve cooperation in the system when the networks obeyed certain rules, but when the networks obeyed different rules, the cooperation disappeared within the system. So think about these two objects. They're both made of carbon. And as we all know, these are allotropes of carbon. And if you take the carbon atoms and you connect them one way, you get graphite, which is soft and dark. And if you connect them another way, you get diamond, which is hard and clear. And then there are two key ideas here. First, these properties of softness and darkness and hardness and clearness do not inhere in the carbon atoms. They're not properties of the carbon atoms, they're properties of the collection of carbon atoms. And second, which properties you get depends on how you connect the carbon atoms to each other. And similarly, the pattern of our connections affects the pattern or the properties of social groups. It's the ties between people that make the whole greater than the sum of its parts. And new properties, maybe even cooperation, infection resistance, and intelligence, may emerge because of the connections, because of the ties between people, and not necessarily because of the people themselves. Actually, I believe that our experience of the world depends on the actual structure of the social ties around us, near and far, and I believe that evolution is shaping the structure and function of networks that all of us create and live in every day. Thank you very much.
Thank you. A gift?
Thank you.
I think we have time maybe for one question. We have one back there. Okay.
Okay. Thank you very much for that. Two questions occur to me. If I can have friends who are male or female, does that make any difference to homophily, heterophily in terms of your networks?
I'm sorry, if you could befriend what?
Well, whether it's heterophily or...
No, I missed the first part of your question. I'm sorry.
Oh, gender. The gender of my friends. I mean, the women
yes.
I might marry, the men I probably won't.
Yes. So we're affirmatively interested in non-reproductive unions. So we have looked at same-sex friendships where there's not a sexual relationship between the individual. We've also looked at discordant sex friendships where there's no sexual relationship between the individuals. And they're actually fascinating patterns. If you're a man and you're straight, who are the women you pick as your friends as compared to who are the women you pick as your potential mates, for reasons that I don't have time to go into right now. I know I didn't give you a fully adequate answer to that question. Come and see me afterwards.
Yeah, come. He's around. Good. Thank you very much.
Thank you so much.
The final talk here is by Peter Holme. He's going to talk about the paradox of importance in network epidemiology. Let's see.
Okay, everybody, can you hear me? Okay, so good morning, everybody, and thanks a lot to thanks to the organizer. It's been a really great conference so far. Thanks to Laszlo and Nicholas for warming up the audience for me. Yeah. So I'm Petter Holme from Sungkyunkwan University in Korea. I'm here to present the project together with Jari Saramäki from Aalto University in Helsinki. And actually, I'm sorry I have to start with a kind of excuse or apology. So I will steal this from Uri Alon, the computational biologist. So he says that when you start as a PhD student, when you're new to science, you think of project as like you have a starting point A and you have a goal B that you can already see, and then you go to B. And maybe like 5% of all projects are just like that, but like 95%, you go from A into a cloud. And you kind of go around in the cloud, and you get out somewhere else. So when we submitted this abstract to this conference, we thought this is one of the rare cases of going A to B, but we're actually still in the cloud. So I'm not going to be able to show you any results today. I think I can give you something that is you want even more than result, which is a good ideas and a good story too. And the good story will be I will present it as a timeline of like influential work for this work. I know there are lots of computer scientists in the audience, and I think you can agree that any kind of timeline for a computational conference should start with the first programmable computer, ENIAC, which was fired up in 1947. So actually the second program to run on ENIAC was the first simulation, for the first program to use random numbers. The code name was Monte Carlo, and that name kind of stuck. It was actually the first program, modern program in the sense that it's a program that you load into the computer with the input data. And important in many ways. Not very soon after, actually, was the first computational epidemiology study. Of course, the computer those days were mostly used by the military. So this is the first simulation project published in an academic journal that I know of. And it was actually a PhD work, which in a thesis presented 1 year early, so very early stuff. Okay, so let's skip the 60s and go to the 70s. This is the core group concept is something that's very important for our work. It was by York, Heathcote, and Nold when they studied the transmission of gonorrhea. They argue that the critical feature governing the dynamics is this very interconnected core of people that are also they are both active and very interconnected. And this is an important concept also because it's one of the few concepts that actually went from theoretical epidemiology to medical epidemiology, along with epidemic thresholds, for example, and a few other concepts. In the 60s the 80s was the birth of network epidemiology. So it's a little bit hard to pick out one paper that started network epidemiology, but I think this is the one with the best story. It's a paper that proved that HIV is a sexually transmitted disease. So they did that by mapping out the sexual contacts between in the early HIV positive in the US. And it has a nice extra story because, it introduced the concept to the English language. Because the PI of the project showed this graph to the first infected patient, and he has number 0 here. His name was Gaetan Dugas. He was a flight attendant, and there are films about him, so you can check it if you're interested. But when he noticed that he has number 0, he started to talk about himself as patient 0, and that kind of stuck in the English language, at least in the 80s. Okay, so the last thing I want to highlight, this is a paper I think is a little bit undercredited by Miriam Kretschmar. I think it's one of the first papers that kind of modern network science in the sense that she has a model to control the network structure. So she tunes the network structure kind of on the x-axis. On the y-axis, she has the dynamic parameters from or output of a disease simulation model. So yeah, maybe worth noticing too that also Miriam Kretschmer is a woman, so is Helen Abbey. So there was some kind of tweets yesterday that this is so male-dominated. At least we have some female pioneers. Also, the Monte Carlo project was coded by Clara von Neumann. Okay, so let's move to our work, and we have to stick to the convention about how to model disease spreading. So that as an emergent phenomena. So it has like two parts of it. There is the first part is the compartmental models, which is to divide people into groups with respect to the disease, and also attach transmission rules. So the groups could be susceptible or infectious, and the transmission rules, like if a susceptible meets an infectious, the susceptible can become infectious. And given some time, she can become susceptible again, defining the SIS model, or recovered, defining the SIR model. Sorry. So this is one aspect of modeling a disease. The second aspect is to the contact patterns. So in this figure, we see our hero girl moving around in her everyday life, meeting different people at different times. So it kind of highlights the two aspects of contact patterns. It's the pairwise interaction, that's the network, and there is the temporal, there is the time dimension. And both, they can be structured in both time and the network, and this can affect disease spreading. So in this work, we'll assume that we can kind of integrate out the time structure, but we also have some work where we didn't do that. So let's go back to the core group idea. So this is an attempt for me to reformulate the definition of a core group verbally. So the kind of bringing together aspects from different, also the medical discipline. So a core group is a group of people that kind of bring a population over an epidemic threshold, even if it wouldn't be if everybody was as the average. Well, this leads to the paradox. Because so this kind of tells us that being a member of a core group is being important for disease spreading. But the core group is very intensely connected. So by definition, if you remove one guy, or say vaccinate one person, or like stop him from having or spreading the disease, it wouldn't matter much. It would be replaced by somebody else in the core group. Yeah, so it kind of tells us that there are different aspects, different facets of the important. And yeah, ultimately, it would, which one that is relevant would actually depend on what kind of specific disease we're thinking about and what kind of intervention scenarios we could afford. It also leads to different measures of what's importance are. Like you can think about the size of an outbreak if a node is the source. This is the, that's the kind of Hernan-Maxes style. You can think about the chance of being infected, which reminds me of the speaker after the coffee breaks in Anaral is work on information spreading and viral marketing. And then, of course, being a member of a core group, or like making a big change if you're vaccinated. This could be two other aspects, ways of thinking about importance. Okay, so to operationalize this, or to make this project, we have to make some more assumptions. So one thing we assume is that the core group can be encoded into a static network structure of a simple graph. We also assume that we can couple this verbal definition to the network by looking at the survival rate of the SIS dynamics. So this is also motivated by this York et al.'s first work. And then, yeah, of course, that the vaccination impact, as I said, is some kind of form of the core group member importance. So in principle, if we can afford to vaccinate many people, we vaccinate the core group and we're done. If we can't afford to vaccinate many people, we can maybe focus on the hubs. If the hubs are outside of the core group, this is our idea. Or maybe in this particular case, we could cut the bridges. So the importance people, if we can afford to infect many, could be different from if we could afford to vaccinate few. But it didn't really work out. So actually, in this setup, the vaccination impact is very correlated with the degree. Well, this is okay. So let's leave that part. There is another angle you can start, or another way to go. If we look at this example network and ask ourselves, like, which one to vaccinate to make the biggest impact, to reduce the size of the largest cluster as much as possible, it will be this one here, if we have only one node. If we have 2 nodes, it's this one, not including the first node. If we have 3 nodes, it's these 3, not including the one at level 2 and 1. And you can kind of continue constructing a network where the important set of nodes, if you vaccinate n, depends on n. Okay, but this, let's leave this project. I just want to mention that Jari and I, we can get out of the cloud sometimes. We have a review paper about temporal networks and a book. So this is like about including time into the network. We have a lot of research. You can I will put the slides online. You can check it up, the references. I wanted to highlight 2 of them. It's going to take 2 minutes or 1.5. So this project is about, like, how can we reduce a temporal network to a static network? And of course, this is something that we really want to do because static networks have this great visual property that it's really easy to use them to discuss stuff. But if we want to, like, add time to it, we're in kind of trouble. So this paper argues that we can, actually the best thing is the simplest thing to do, and to take a time window and just aggregate the contacts in between. But we have to be careful of what's the beginning and the end of this time window. And the last thing I wanted to show is this project. It's kind of addresses, like, how can we simplify, how can we think about temporal networks in a simple way? The one way of doing it is to think of it as a static network with a dynamic system generating the contacts on top of the links. But we argue for disease spreading on the empirical data set. A better way to think of it is to think of the first contact as the beginning of a relationship and the last contact as the end of a relationship, and then we don't really care what happens in between. So with that, I want to thank you all for listening. Thanks.
Thank you. We have time for maybe one question. One question? No? Nothing obvious? Okay. So coffee break for half an hour. And remember, there are free books down at the registration. Thanks. Good. Thank you.
Should have brought my hard copy so I could get it off.